thanks so much for a great presentation. Thanks, Hamid, for organizing the webinar. For those who don't know me, I'm Alex Opitz. I'm Associate Professor in Biomedical Engineering at the University of Minnesota. Next slide. So we're gonna talk about non-invasive brain stimulation. So mostly focus on electric stimulation and transcranial magnetic stimulation that both create an electric field in the brain. Next slide. Next slide. Yeah, so if we talk about non-invasive brain stimulation, we have sometimes a bit of a problem for black box. So we apply something on the outside, outside of the head, say we attach electrodes on the scalp level. But what we're really interested in, what's happening on the level of the brain. So how do those electric fields affect neurons? Is there physiological response at all? Where do those where those currents go? So this is a tough problem to solve, also in particular if you're doing non-invasive research in humans. And so animal models can really help elucidating those basic mechanisms a lot. Next slide. So, but creating animal models that will be truly translational for non-invasive brain stimulation is not an easy task. So at the end of the day, we want to treat humans with those methods. And so what we're studying in animals should be translated to humans. And so there are just some challenges just from the basic physics of it, because if you look at those slides and some head models of a human at the right and a non-human primate and the red, you see the first big differences in just like oval head size, muscle, skull composition, and so on. And they will all have a big influence on what the electric field will be like at the level of the brain if you apply non-invasive brain stimulation. Next slide. And so we were really trying to characterize that in more detail. And so we have a paper led by Ivan Alexeychuk, where we just looked what would be the electric field in the brain be from different non-invasive brain stimulation protocols. The examples that you see here on the slide is from electric field from transcranial electric stimulation. If you just would apply, say, 1 milliamp stimulation at the scalp level, and what you see there is like a range of a factor of, say, 60 from the mouse or rat model to a human. And so non-human primate is somewhere in between. It's still somewhat higher than a human, but already quite close. And so this characterization allows them also to, say, develop protocols or apply your dosing in a way to get translatable protocols. Next slide. And so one study where we were looking at a dose response of transcranial alternating current stimulation in non-human primates, we looked at the effect of different stimulation intensities on spike timing. One big advantage with those invasive recordings is you can directly record single units or spikes. That's a signal that you can recover even in presence of stimulation artifacts. Next slide. And so what we found there was that the level of alignment of spikes with the stimulation phase of the external oscillatory currents was like increasingly aligned with the peak of the stimulation. So you see all those dots as a occurrence of a spike. And then you see like some more peaks towards then, say, the center of it. That's where like the peak of that oscillation happens. And so with increasing electric field strength, they also found more spiking, more neurons entrained to that oscillation. Next slide. So yeah, this was a demonstration in TACS, and just like highlight some current work we're doing. We're also using now for transcranial magnetic stimulation with stereo EEG electrode recordings in non-human primates, also investigating dose responses with TMS EEG evoked responses, and there we also have to see dose response with respect to TMS intensity. Next slide. Yeah, just highlight collaborate. I think that helped doing this work. And next slide. And our thanks to the lab and funding agency. Next slide. And just the final thing, we have open positions of interest in any of the work. Yeah, feel free to reach out to me. All right, thank you.